All right, so throughout your degree, you've probably had the standard model of cell division drilled into you, right? One cell becomes two, nice and neat. It's one of the fundamental rules of biology. But what if I told you that for some organisms, that rule is, shall we say, more of a suggestion? We're about to dive into the absolutely wild world of parasitic protozoa. These are organisms that have spent literally a billion years just ignoring our textbooks. And in doing so, they give us a masterclass in just how creative evolution can be.
So here's the game plan for today. First, we'll quickly recap the official cell division rulebook, just to set the stage. Then we'll get to the good stuff, how parasites just completely break those rules. We'll look at two amazing case studies, the super precise duplication of trypanosomes, and then the truly bizarre Russian doll strategy you see in Apica complexans. And to wrap it all up, we'll talk about why all this weird biology is actually a huge deal for developing new drugs.
Okay, let's kick things off. Section 1, the Cell Division Rulebook. You know, the orthodox model. Before we can really get into the weird and wonderful, we've got to have a solid baseline of what normal looks like.
And here it is, in all its glory. This is that canonical pathway we all know and love. It's just so orderly, isn't it? You see the spindle assembly, then that classic contractile actomyosin ring forms, pinching in to create the cleavage furrow. It creates that intercellular bridge and then, boom, abscission. You're left with two perfect, genetically identical daughter cells. It's elegant, it's predictable, it's the gold standard that you'll find in any cell biology textbook.
All right, now for part 2, parasites breaking the rules. And this is where things start to get, well, a bit weird. We're going to dive into why they throw the rulebook out. Because, and this is key, their strategies aren't just random chaos. They're incredible, highly tuned adaptations, all driven by the immense pressure of having to survive inside another living thing.
This phylogenetic tree, honestly, it says it all. It really shows you why these organisms are so different from us. See, we're animals, right? We're tucked away in our own little supergroup, the Opisthoconta. But the parasites we're talking about today, like the kinetoplastids and apicomplexans, they're in completely different branches of the tree of life. We're talking about a separation of over 1 billion years of evolution. Think about that. That is an unimaginable amount of time to come up with completely different ways to solve the same fundamental problem. How do you make more of yourself? So this is the take-home message here. For a parasite, replication isn't just a biological process. It is a high-stakes game of survival. It's not just about making a copy, it's about making copies fast. It's about overwhelming the host's immune system, generating massive numbers, and doing it all in what is, let's face it, a pretty hostile environment. So these bizarre methods we're about to see, they're not sloppy. They're hyper-specialized, incredibly effective strategies for winning that game.
Okay, let's dive into our first case study, trypanosomes. And the big problem for a trypanosome is a mechanical one. I mean, how on earth do you make a perfect copy of a cell that's so complex and, well, so weirdly shaped?
I mean, just look at these images of Trypanosoma brucei. This is not your simple spherical cell. It's got this incredibly defined, long, elegant shape. And it has a single flagellum that pops out of a flagellar pocket, runs all the way down the cell body, and it's attached, creating that classic undulating membrane. So the biological puzzle is just massive. How do you split this down the middle and make sure both new cells get a perfect working copy of every single one of those bits and pieces, all in exactly the right place? It's a huge challenge.
Well, the answer is just beautiful. It's this incredibly precise, almost factory-like assembly line. It's all about strict timing and placement. So check this out. First, a new flagellum and its anchor, the basal body, get duplicated. Then, the kinetoplast, that's their unique bundle of mitochondrial DNA, divides. You know what's wild? The nuclear mitosis, the thing we usually think of as the main event, happens much later in the process. And then finally, cytokinesis itself doesn't just pinch in the middle. Nope. It starts at the front, at the flagellar end, and basically zips the cell in two along its long axis. It's this perfectly choreographed dance to make sure everything ends up where it's supposed to.
Okay, moving on to section 4. And if you thought the trypanosomes were clever, you haven't seen anything yet. We're talking about the apicomplexans, and their strategy is, well, the best way to describe it is the Russian doll strategy. You're about to see why that analogy is so perfect. And here we have it, the matryoshka doll. It is the absolute perfect analogy for what's going on here. Because instead of dividing from the outside in, with a cleavage furrow pinching the cell,
Apicomplex and parasites like Toxoplasma and the malaria parasite Plasmodium do something that's just completely backwards. They build brand new daughter cells inside the mother cell, literally like one doll inside another.
And this diagram really shows you how that analogy plays out biologically. This is Toxoplasma doing something called endogeny. Those daughter cells, which you can see in yellow, are literally being built on an internal protein scaffold. It's called the intermembrane complex, or IMC. All this is happening inside the mother cell's own plasma membrane. The mother cell basically turns itself into a factory for its own offspring, and when they're ready, they burst out, consuming the mother cell from the inside out. It's incredible.
But what happens when you need to make more than just two? Well, parasites have scaled up this internal budding strategy for maximum damage. The process is called schizogony, and it is the absolute weapon of choice for Plasmodium, the parasite that causes malaria. What happens here is just mind-blowing. Instead of making two daughters, the nucleus just divides again and again and again, multiple rounds of mitosis with no cell division at all. Then, once you have a whole collection of nuclei, a brand new daughter parasite buds around each one, all at the same time, all inside one single host red blood cell.
And this is the end result. It's both spectacular and, frankly, terrifying. What you're looking at is a mature schizont inside a host cell. It's basically a biological time bomb. That beautiful rosette pattern is actually dozens of newly formed daughter parasites. We call them merozoites. Each one is a fully formed infectious agent, just waiting for the signal to burst out and invade a whole new set of red blood cells. This is how malaria gets out of control so fast. It's a strategy of pure, explosive amplification.
Okay, so we've seen some absolutely incredible biology, but you might be asking, so what? Why does any of this matter in the real world? Well, our final section is all about that. It connects all this evolutionary weirdness directly to drug development. And honestly, this might be the most important part of the whole story.
So let's just recap the key points here. Parasites have this incredible flexibility in cell division. We've seen internal budding or endodiogeny and that explosive schizogony. Their cytokinesis is often budding, not the furrowing we see in our cells. But the really critical difference, the one that matters most, is at the molecular level. The actual proteins that drive and regulate this whole process, they are fundamentally different from ours. Many are totally unique to the parasite. And that is the key.
And that brings us to the biggest takeaway, the most powerful idea of all. This massive evolutionary divergence, it's actually our greatest weapon against them. Because their replication machinery is so profoundly different from ours, it gives us a beautiful set of unique molecular targets. It means we can design drugs that go in and specifically smash their cell division machinery, maybe block their weird budding process, or inhibit one of their unique regulatory proteins, while completely leaving our own cells alone. That's the holy grail of antimicrobial therapy, right? High specificity and low toxicity to the host.
So I want to leave you with one final thought to chew on. We've just seen how these parasites have completely and utterly reinvented one of the most fundamental processes in all of biology. They've shown us that there are other, very successful ways to be a eukaryote. So if they can so radically rewrite a rule as basic as cell division, what other biological rules that we take for granted are actually, in the grand creative scheme of evolution, just suggestions?